Now our next group is Ascomycetes. Now see the fungal hyphae of Ascomycetes. It is having septa and it is having branches. So we can say septate branched fungi, and commonly it is called sac fungi. It can be asked as a separate question in 1 mark. It is saprophytic, act as decomposer, it can be parasitic, it can be unicellular or it can be multicellular. So we are saying that it is branched, it is septate fungi, commonly called as sac fungi. It is saprophytic, decomposers, parasitic, unicellular or multicellular. So we will study one example of unicellular and multicellular. First I am coming towards the unicellular fungi. See, unicellular fungi example is yeast. In yeast, when we are talking about the yeast, we have to study the structure, function as well as reproduction. In yeast, cell wall is made up of chitin. We know that. In fungi, cell wall is made up of chitin. Along with chitin, what else is present? Phosphoric acid and glycogen. Yeast is secreting an enzyme called zymase, you should underline, which converts complex sugar to simple sugar. That is why we are saying that yeast is used for fermentation. So what yeast is doing? Yeast is having an enzyme called zymase which convert complex sugar to simple sugar. This is the internal structure or the morphological structure of the yeast. It's a unicellular fungi. And reproduction by budding and fission. Budding, you all know that very well, you have studied in 10th class also. Yeast reproduces by budding. Now when we are talking about the fission, you have not studied about the fission of the yeast. Cytoplasmic structure elongates first. That means plasmogamy. You have studied union of protoplasm, but here we are saying that protoplasmic division takes place first, then nuclear division, and that is called as fission. So after that, when we are talking about the sexual reproduction takes place by 3 steps which you have studied: plasmogamy, karyogamy and meiosis. You have studied earlier plasmogamy, fusion of protoplasm, karyogamy, fusion of nucleus, and meiosis means division of zygote, division and differentiation of zygote. In this way, yeast have been multiplied. So this is all about the unicellular fungi. Now come to the multicellular fungi example, that is called as penicillium. Now see the structure of penicillium. Again we are saying that it is a septate hyphae. And when we are studying the structure of penicillium, you all know that penicillium is having, it comes under higher fungi. So it is having conidia. Asexual reproduction by conidiophores. These fruiting bodies we can see, these are called conidiophores and they are producing these rounded structures. These are called as conidia. So asexual reproduction by conidiophores, and suppose this is hyphae, this is producing fruiting bodies, conidiophores, they produce conidia, and conidia again germinate and form the hyphae. Then hyphae will again form conidiophores and conidia. This is the cycle of asexual reproduction. When we are talking about the sexual reproduction takes place by ascospores. So ascospores are the structure of the penicillium, or we can say that Ascomycetes, and endogenously they are produced in asci. Endogenously and exogenously 2 structures are there. So spores are produced inside or spores are produced outside. So they are endogenously produced and arranged in fruiting bodies called as ascocarps. So you should be familiar with the word ascospores, asci, ascocarps. These are the structures which should be used in the asexual reproduction, in sexual reproduction in case of penicillium. So this is all about the yeast and penicillium, about Ascomycetes. Now come to next group, that is called Basidiomycetes. When we come to the Basidiomycetes, now in this group comes mushrooms, bracket fungi or puffballs. Examples are mushrooms, bracket fungi or puffballs. You might be familiar with the mushroom structure of mushrooms. It is found in soil, logs of wood, tree stumps, in the living plant bodies as a parasites. Example, rust and smuts. As a parasite, examples are rust and smuts. If you want to see the structure, again you can see septa are present, branches are present, so we can say branched and septate. Vegetative reproduction takes place by fragmentation. Asexual reproduction takes place by spores, which are not found. So we can say that asexual reproduction, spores are not found. I put a cross over here. That means asexual reproduction cannot take place through spores. So it is missing. When we are talking about the sexual reproduction, sex organs are absent, but sexual reproduction takes place. How sexual reproduction takes place if sex organs are absent? Now this is the method. Two mycelium with different strains like positive and negative. Positive can act as male, negative can act as female, fuse together, and in this way reproduction takes place. So we can say that suppose this is positive strain, this is negative strain, both the mycelium close together and they form sexually reproducing body, 2N form, and some types, or we can say that it is a same type dikaryon stage. Now see, in this we will continue with the Basidiomycetes. Like in last video you were studying that positive and negative strain fuse together, and positive and negative when fused together they form the protoplasm. Fusion of protoplasm takes place and that stage is called as plasmogamy. So after plasmogamy what will happen? They form the structure and that is called as basidium. That is called basidium. After that karyogamy takes place, that means fusion of nucleus, and continue with the karyogamy, meiosis takes place. So plasmogamy, karyogamy and then meiosis. After that they form 4 basidiospores exogenously. I use the word exogenously. That means the zygote like structure is formed. Over that 4 basidiospore have been formed. So after meiosis 4 basidiospores have been formed, and this is the process of sexual reproduction. And examples are Agaricus, Astilago, Puccinia. They can decompose cellulose and lignin. So they are the good mineral adding agents in the soil. So they act as a decomposers also. Now come to next group, that is called as Deuteromycetes. When we are talking about the Deuteromycetes, you might remember that I use the word fungi imperfecti for this. These fungi are commonly called as imperfect fungi because sexual reproduction is absent or not known. They are saprophytic, parasitic, large number of them are decomposers. So we can say that they help in the mineral cycling, and their most of the characters are similar to Ascomycetes and Basidiomycetes. They are having the septa as well as branch in the hyphae, so branched and septate. Examples are Alternaria, and you can see the spelling, a new word for you, Colectotrichum and Trichoderma. 3 examples I have given. and reproduction takes place by conidia. Conidia are circular structures like this without flagella. These are conidia. You have seen in penicillium also. This group have the common characteristic of Ascomycetes and Basidiomycetes. So this is all about the Deuteromycetes.